Okay, so we're going to talk about cellular respiration today in terms of substrates, products, relative amount of ATP, and location in the cell. Okay, so there's, depending on how you're counting, like 3 or 4 main processes going on here. So those are glycolysis, prep step, TCA, electron transport. Okay, and there's 2 main locations in eukaryotes, which is going to be the subject of my talk today. For prokaryotes, it's the same idea, but instead of having the mitochondria involved, so this would be the mitochondrial matrix over here. So prokaryotes don't have mitochondria. Instead, the same process would take place in the periplasmic space. And that's the space between the cell membrane and the cell wall. So here we've got the mitochondrial membranes. This is the outer membrane. This is the inner membrane of our mitochondria. Okay. And on the innermost part is called the mitochondrial matrix. Okay, so over here we've got the cytosol, that liquidy part in the cells. All cells have this, and also all cells have the first step of this, which is glycolysis. And not to complicate this, this literally means sugar cutting, and that's when you take glucose, which is 6 carbons, and through a series of like 9 enzymes, you make pyruvate. And the trick is, this is a C6 compound, this is a C3 compound. We have to have the same number of carbons in each. So 2 times 3 is 6. So 6 carbons starting with, 6 carbons ending with. Okay, so that's the first step. In glycolysis, we have a couple other things happening. So glucose is being converted to pyruvate through 9 amazing enzymes. We also have NAD plus being converted to NADH in this process. This is a coenzyme. This is very important for cells, and in fermentation we have to regenerate this NAD plus. So in order to do that, the fermentation takes this NADH, and because this is a good reducing agent, it has this H on it, it reduces pyruvate in order to make a variety of different compounds, and that regenerates the NAD+. So there's this reduction, and then fermentation does an oxidation back. We're not doing that here. Here we've got electron carriers, so we're going to see this enter the mitochondrial matrix later. Okay, this pyruvate, this is the last time it's in the cytosol, because then it's going to be transported into the matrix for the next step, which is the preparatory step. This is also called pyruvate oxidation. So basically this pyruvate is then oxidized to acetyl-CoA. And when this happens, 1 molecule of CO2 is released. And also, we get a molecule of NADH, which we can use here. So in fermentation, it was important to oxidize that back. Here, we're going to use it for energy first and then oxidize it back. Okay, so I'm going to go ahead and scroll down a little bit, give myself a little bit more room here. Okay. So we're still in the mitochondrial matrix. This pyruvate gets transported, transformed to acetyl-CoA through that second step in that goldenrod color. Second step is called the prep step. This only happens if you're undergoing respiration. If you don't have enough oxygen, then you're going to just do fermentation. Okie dokie. So then next, we're going to do TCA, the tricarboxylic acid cycle, also known as the Krebs cycle. I'm just going to draw it like this. This is a series of enzymes again, a whole bunch of different enzymes going on in here. This makes a bunch of really important intermediates for the cell, but some other things that it makes includes, oh, you know, I didn't mention ATP before, but of course we made ATP. So ATP, and we end up with 2 ATP from here, no ATP there, and here we're going to make 1 ATP per cycle. We're also going to make NADH, our high energy reduced electron carrier. We're also going to make FADH2. That doesn't go too far. And we're also going to make a whole bunch of CO2. So that's everything that comes out of here. Okay. And then when this is all done, we still have, so total we've made in terms of ATP, we've got 2 from glycolysis. We've got 1 per cycle here. We can do 2 turns. We've got 4 total ATP, which is super lame. This is a lot of work to just get 4 ATP. So what these cells do, when oxygen is present, this is why oxygen is such a huge advantage, is that we can use it as a terminal electron carrier and undergo the 4th step. So 3 was TCA. The 4th step here is called electron transport, also known as oxidative phosphorylation. So I call it ETC, electron transport chain, etc., etc. Okay, so we've got these high energy electron carrying molecules pretty much all over the place. Wherever you see an H, you know that's carrying electrons. Okay, so these are going to be transported to the membranes. And here we've got a bunch of membrane-bound proton pumps. And at the very end here, we have ATP synthase. It kind of looks like a lollipop. Okay? So, what this is going to do is it's going to collect all of the electrons from all these different things and return them as NAD+. So it's going to steal the electrons off of the NADH. Everything's going to feed into here. NADH is going to go up there. FADH2 is going to go up there. And basically what's happening is it's dropping off its electron. And every time the electron moves from a high energy to a low energy, it loses a little bit of energy. And each one of these enzymes is a proton pump. So that means it takes a proton, an H+, and moves it into this membrane space. So this is why this has to take place in the mitochondria. This is why it's important that the mitochondria has multiple layers. And in prokaryotes, like we mentioned, this would be the periplasmic space, the space between the cell wall and the cell membrane, because they don't have organelles. So we end up with a high concentration of H+ right here. And then in order to make the ATP, we can take things from a high concentration and move them to a low concentration just through diffusion. So that's exactly what happens. We don't have to put in energy for diffusion. So our huge ATP synthase enzyme is going to take that huge concentration of H+, use that like a water wheel, and synthesize ATP. So ADP goes in, an inorganic phosphate, and what comes out is the biggest amount of ATP that you could possibly imagine. So we get about 32 to 34 ATP. Okay, so which one of these 4 has the most ATP? That's electron transport. Which one of these occur in the cytosol? That's glycolysis. Everything else is in the mitochondrial matrix or embedded in the mitochondrial membrane. Okay, so once this electron makes it all the way down, it still has to go somewhere, and that final the final place that it goes is called the terminal electron acceptor. And for organisms like us who breathe oxygen, oxygens are that terminal electron acceptor for us. So it's going to accept that electron. When it does that, it becomes reduced, and it makes a molecule of water. So this is reduced, has an H, has gained electrons, etc., etc. So this water, I show it leaving, but it could stay on this side. It could still be in the matrix. Okay. And that's what I've got. Those are the 4 stages of cellular respiration. It doesn't have to be oxygen as the terminal electron acceptor, but in our case it is, so I think it's a great one. Whatever the terminal electron acceptor is will become reduced. Electron transports the highest amount of ATP. TCA is low. Prep step is the lowest. It doesn't make any ATP. So glucose TCA in the middle there. And then you can see where everything is made. So I hope that's a little bit clearer. Thank you for listening.